Hi everyone. Hi Michael. So we are good to go. We are recording, and today we'll be having Michael. Hi Michael. Am I audible?
You're audible. Hello.
Yes. You're audible. Okay.
Great.
Yeah. So are we good to go?
Yeah, I think so.
Okay. So give us a moment. We'll start the live on Facebook, and then we'll go ahead. Right?
Sounds good.
Okay.
Hello everyone, and today we have Michael with us, and he's the 2021 Young Researcher. He got the 2021 Young Researcher Award from GBIF, and he is currently doing his PhD at the University of Florida, and he is a graduate. He was a graduate assistant actually previously in the Florida Natural History Museum, and he has a diverse field of work. He has worked on different citizen science initiatives. And he is very good at climate change biologist as a climate change biologist. And we are glad to have you here among us to start this initiative of the series of talks with you. So today's topic is overwintering strategy impacts on butterfly phenology and abundance. So we can start it now. If we, Michael, if you have any, if you are ready, then we can start.
Okay, that sounds great. I'll share my screen, and hopefully then it goes to the right stuff. So I am sharing now. And do you see the slides, or do you see presenter view?
Yeah, and the presenter is now showing. Yeah.
And do you see the full screen, or is it showing like my notes?
Yeah, full screen. Yeah.
Okay, perfect. I think we're good to go then. Thanks for that introduction. I'm happy to be here, and looking forward to this talk and all the questions. So thanks again, and I will be talking about butterflies, particularly their overwintering stage, to understand the ecological consequences of global change and macroecological scales. First, I'm going to take some time to discuss how I got into biodiversity science, and then give a brief overview about the types of research that I do with my work. And then in the third part of my talk, kind of the last half, I'm going to be focused on a specific case study looking at one research project. So as a kid, I always enjoyed being outside. I liked catching frogs and crayfish in the local stream, and also going around my house looking at all the bugs and plants that were around. But I wasn't really too interested in thinking about careers or getting a job. And I had never heard of a research career. So it wasn't until undergrad, during a study abroad experience with the School for Field Studies in the Australian wet tropics, in which I was first introduced to field ecology. I thought it was the coolest thing ever that people had jobs where they could be outside, measure trees, or watch flying squirrels, look at bugs, and they got paid to discover new insights about the natural world. I just thought that was the coolest thing. And this led me to work as a field technician on a few jobs where I was beginning to learn a variety of field techniques. Pretty quickly, I wanted to take a larger role in working with the data that I was helping to collect. And I wanted to use these data to produce research that could inform conservation. So I really started by doing field work, and then I wanted to use the data too. So thus, I started a master's program with Anna Monfils at Central Michigan University, where I worked on projects to inform the conservation of a critically endangered butterfly species, the Powsheik skipperling. As demonstrated by this map on the left side, the Powsheik skipperling used to be found at hundreds of sites throughout the Great Plains of the US, but its range drastically shrunk over recent years, and it's now found at only a few sites in Michigan and Manitoba, Canada. During my master's, I took approaches from both field ecology and data science to learn more about the ecology of these species. A lot is just unknown about this species, including its preferred host plants. So we were in the field looking at what host plants the species was laying its eggs on. We were looking at preferred nectar hosts, the actual seasonal timing of its flight period. And then we tried to use historical records to piece together potential drivers of decline. Some of the stuff we found of actually what nectar is available is Dasyphora fruticosa, this scrubby cinquefoil, and Rudbeckia hirta were some of the most abundant nectar sources, and they also just nectared on those. So it, in a lot of ways, was a nectar generalist. And the flight period showed that the first Powsheik would be flying around the June 25th, and the last one was around the mid of July. So its flight period was only lasting just under like a month. It is univoltine and a really short flight period, with peaks arriving around July 4th. And then we also did a population density estimate by looking at distance sampling using a technique where birders actually use this a lot, where they do point counts and how far away the bird is from them. You can use some mathematics to figure out actual population density. And we use that with butterflies. So at standardized point counts, we would measure the distance away that we saw butterflies to get a density. And given that we knew the actual suitable habitat in these populations of prairie fens, we estimated that all of the butterflies left in Michigan, which is the stronghold of its population, was under 500, with confidence intervals between 250 and 464. So this is a critically endangered butterfly that globally has probably less than 500 in the wild. And while working with the Powsheik, I realized I really enjoyed the computational aspects of the work. Not only did I like being in the field, but I really liked the computational work and that I was pretty good at it. So additionally, while I was doing the master's project, the papers that I was reading that I was most excited about were using large and messy data sets to draw ecological conclusions for many species. So far for my PhD, I moved into work in a biodiversity informatics lab with Rob Gorelnik at the University of Florida. So now I'm going to quickly go over some of the breadth of my research, focusing on three main areas, and those being macroecology, global change biology, and documenting and overcoming biases and incidental data to scale biodiversity science. Macroecology is the study of the mechanisms underlying general patterns of ecology across large spatial scales. I have worked in macroecology lens, looking at species richness and endemism patterns, as well as phylogenetic richness and endemism patterns of insects at the largest scales possible. So this is looking at the phylogenetic diversity on the left side of butterflies, and then plants is actually the one on the lower right next to the phylogenetic diversity of butterflies. And there are patterns that match in some ways, but are different in others between butterflies and plants. And actually the richness patterns across North America of dragonflies is shown on the right. And dragonflies show a east to west gradient, where in the east, there's a lot more fresh water. So as you'd expect, a species that needs fresh water are going to be more rich in those areas, but butterflies, on the other hand, show that a north to south within the tropics, there is many more species of butterflies in the tropics, and also more relative richness in the west with more complex habitat. My work also looks at global change biology, which is the study of how environmental change, such as climate change, land use change, invasive species, and urbanizations impact biological systems. Much of my work has really focused on phenology, or the seasonal timing of events. So in one study, I looked at the phenological timing of adult activity in insects across 101 insect species, most of which were butterflies and moths, and dragonflies and damselflies that compiled most of the species. And we looked at how long their flight periods occurred across a temperature gradient. And we sorted that out, conditioned that by life history traits. And what we found was that species in warmer areas had longer flight periods if they were detritivores. So if they ate detritus as larval, potentially because detritus just has much more seasonal availability in warmer areas. And we found freshwater had the species with immature habitat in freshwater, such as those dragon and damselflies, some beetles, they had longer durations in warmer areas. So they were flying longer as adults in warmer areas, but species that had immature habitats, spent their larval stages underground, basically had the same adult flight periods across a temperature gradient. So whether they're in a cool or warm region, they always had the same flight periods. This has implications giving climate change about how long these species may be active and when their ecosystem services will be available in a changing climate. But both my macroecological and global change work really relies on developing methods to integrate incidental data to scale biodiversity science. And there are a number of reasons why I think it's important to develop these methods that I'll quickly outline in the next few slides. So incidental data, that is data that is unstructured, not using formal surveys, but instead is often used by community scientists or people collecting from museums, where the data itself is totally opportunistic, and oftentimes the only information you have is that a species occurred at this location, this time, and it is this species. And that's all you have. You don't have information of how much, how hard people were looking for stuff, if they recorded everything they saw, or just that one species. So there is a lot missing in these data, but it has tremendous coverage. So iNaturalist represents one of these incidental data sources. And I'll talk about iNaturalist for the next few slides. iNaturalist data has tremendous coverage, really global coverage, and it's increasing all over the place. iNaturalist has over 100 million observations, and it's continuing to grow. So each year there are about a doubling. So more and more records are coming in every single year. And these data are being expertly curated by taxonomic experts volunteering their time to share their global expertise. This plot shows how ideas may be from one area, but they are IDing observations from organisms across the globe. This expertise has led to really well curated data sets. I know of example studies in snakes and butterflies, finding that about 99% of observations are correctly ID'd. These data are allowing questions to be asked at scales previously unattainable from more structured data sources. The right panel figure shows the location of records available to examine the flowering timing of yucca. The red dots, which you can hardly see, are the National Ecological Observation Network. So this network of standardized sites throughout the United States with yucca observations. The blue dots are observations posted to the National Phenology Network, another semi-structured to structured phenology network, and the yellow dots are iNaturalist records. So as you can see, compared to these more structured data, iNaturalist has greater spatial coverage. And because each observation on iNaturalist is tied to a photograph, these observations can provide evidence of extremely rare species and behaviors. For example, the first record of a female Australian barkhopper since its description in 1887 was recorded on iNaturalist. And it's only one of 5 records since the holotype was collected, was posted on iNat. And there's also the only known photograph and the only known georeference record anywhere of the species of non-biting midge in the bottom right. It was recorded on iNaturalist. So it has been described before, but the type specimen did not have a georeference record. And now the only georeference record is from iNaturalist. Of course, though, iNaturalist records are incidental data, like I was saying, and they don't provide information about how the data were collected or any information about survey effort. One vignette highlighting this problem is if I were to use iNaturalist to examine the relative abundance of cardinal color morphs at the University of Florida's campus, you will see that most of the cardinal observations are of yellow cardinals, but I can state with high confidence that this was just one individual cardinal getting repeatedly photographed by exuberant birders, and red cardinals are indeed the dominant color morph. Obviously, these data present major challenges to examining questions related to abundance. There's also temporal and spatial biases, so effort is not consistent across a year or in locations. There's more observations that are posted on the weekend, for example. The City Nature Challenge every year creates this huge spike in observations, and most of the sampling is in developed areas. And that is way more than you would expect by chance, given how much urban areas actually take up on the landscape. So a lot of my time and research is actually spent because there is so many data points through incidental data, such as iNaturalist and museum records, that I think these can be super important and useful to research and creating large global change studies. But obviously these biases must be accounted for. So that brings us to the main vignette of a research study that I want to be focusing on today, and that's using museum records to look at the phenological responses of Lepidoptera and how those are conditioned by overwintering strategy. And this is work that was done with collaborators listed below: Elise Larson, Van Shirey, Daizhong Li, and Rob Grant. Shifts in phenology, that is the timing of seasonal biological events, are among the most noticeable impacts of human-caused global change. Impacts are shifting, organisms are shifting their phenologies earlier in a warming climate across the globe and across the tree of life. Recently, researchers are attempting to move beyond simply documenting changes in phenology and are examining more complex phenological responses to better predict responses to future climate changes, urbanization changes, and other global changes. Often, phenological studies are limited to examining only single or a few species. The studies that may include many species are often limited to only occurring at a few locations. So these are very localized studies if there are many years. And what I wanted to do in this study is across many years, so a really long time frame, look at how does boltonism, that's how many broods a butterfly may have in a season, and the overwintering strategy, how do these traits drive differential onset and termination dynamics over time? We know that how many broods a butterfly has in a season and the overwintering strategy is important to when butterflies start flying and when they stop flying on the landscape. But what is less known is over time, how are these species shifting their phenology? So we also know that overall butterfly species are flying earlier due to warming climates, but do these shifts, the phenological sensitivity, are they different across different overwintering strategies? But it's really hard to get at those questions using standardized data because we don't have a long history of observations. Natural history collections are a powerful resource to understand longer-term trends. Natural history collections thread the needle of offering vast taxonomic and temporal coverage and are a resource that is often underutilized by ecologists. But like we said earlier about all that naturalist data, museum data are also messy and biased. For example, these data are unstructured, meaning that these data were not collected using rigorous protocols designed to meet well-defined objectives. Sampling biases also abound, such as often specimen data are collected near city or roads. And additionally, the effort put into collecting is hard for the data practitioner to tease out, and these efforts can change through time and space. There are also reporting preferences, with showy flowers, for example, being collected more than difficult to ID sedges. On top of all these challenges, integrating these data across multiple host institutions and aggregators present their own challenges of how to make these data fair, or findable, accessible, interoperable, and reproducible. These data biases have also changed through time. In this study led by Van Shirey, that Vijay Bharvi and Rob Grolnick were also co-authors on, we found inventory completeness of North American butterflies. So inventory completeness being the number of species collected in an area over the number of species expected to be in an area to be overall greater in more urban areas, suggesting an urban bias in museum samples. However, this urban bias in sampling has been decreasing through time, meaning that we know there's biases in these data, but you can't even assume these biases remain constant over. So given these challenges, can we generate accurate phenology estimates from incidental data? I'm going to take you through a case study where we showcase and document best practices for leveraging natural history collections in phenological studies. Although our case study focuses on the flight periods of adult Lepidoptera, these best practices should be applicable to diversity of phenological responses. Our case study focuses on Lepidoptera, and we had a total of 265 species, of which most were butterflies, but we also had quite a few moths in our data set. Nymphalidae had the greatest number of species in our data set. So nymphalidae being your brush-footed butterflies, such as the checker spot on the upper left, or satyrs, browns shown on the bottom right. Aribidae had the second most amount of species, that's tiger moths. Then Hesperidae had the third most by a family included in our data set. That would be your skippers. And Pieridae, Lycaenids, those are going to be your next most represented butterfly groups. In total, our study looked at the years from 1902 to 2016. So we had 114 years included in our data, where we gathered annual temperature, annual precipitation, and temperature seasonality and precipitation seasonality values. And that covered 104 grids across North America. Previous efforts have been made to make phenology estimates more accurate, but guidelines to assist researchers along an entire project are less common. Here, I'm going to quickly detail the workflow to support best practices in using digital natural history collections data in phenological research. But if you want to know more, I'm happy to announce that this paper just got published this week, actually, in Functional Ecology. And in the next few slides, I'll go through these this methodological checklist that we outline in the paper and discuss the need to do these steps while briefly explaining how we implemented this step of checklist for our butterfly case study. First, we download our digital museum data. For this study, we gathered data from GBIF, iDigBio, and SCAN. We then harmonized taxonomy to generate a standardized list of species for use in analysis. This is especially important if the specimen records were downloaded from a source without a backbone taxonomy, or if multiple data sources are being combined. Taxonomic standardization is a critical challenge as taxonomic names change, and there is no guarantee that data aggregators are either up to date with these changes. In nearly all cases, synonyms must be harmonized to a standardized species list to reduce potential for pseudo-replication. We found that doing so removed 95 synonyms out of the 360 species names from our raw data set that had enough data to estimate phenology. Like any project, data cleaning is a necessary and important step. However, unlike traditional ecological projects, these data sets will have hundreds of thousands to millions of records, and manual cleaning is error-prone and unfeasible. So in this study, we did a variety of data cleaning, but here I'll focus on removing records that were duplicated across data sets and had imprecise records. Duplicate records were records that were collected at the same place at the same time and by the same collector. After removing those records, our data set primarily consisted of GBIF and SCAN records. Next, we removed records that were collected on the first day of the month because the specimen had labels that said collected in June 1973, for example. So it was known it was collected in June, it was known it was collected in 1973, but they didn't know on what date. And in a lot of databases, that defaults to having a date of June 1st, 1973. So this leads to biases where, as you can see in panel C, you have way more records occurring on the first day of the month than would be expected. So we just removed all of the records that occurred on the first day of the month to make sure we had precise date records in our data set. Selecting the spatial grain is also important, and that's because you want to make sure that you have enough data to create robust estimates, but you also want to still have a grain at a size that is meaningful from a biological perspective. Appropriate scales will depend on data densities, the life history of the group of interest, and the spatial and temporal scale which predictor variables operate. While finer grain estimates are often preferable to localized processes driving phenology, these are impractical given the sparseness of natural history collection data. In this case, given the focus on broad continental scale patterns of climate predictors that are highly spatially autocorrelated, coarse grain cells were an acceptable trade-off for upping the number of phenometrics we could estimate. However, studies incorporating land use change may need to sacrifice quantity in order to model these more localized phenomena at finer scales. Deciding on data requirements is a challenge that is still open in the literature. Deciding on minimum data requirements requires necessary, or deciding on minimum data requirements that are necessary for fitting phenometrics is also a key trade-off between number of estimates versus their reliability. In a recent study, we showcased that the minimum data points needed to accurately estimate phenometrics depends on the characteristics of the species. Multiple broods per year and long flight periods require more data points to accurately estimate phenophases. Broadly, there is no one-size-fits-all approach, and minimum data requirements should be carefully selected with regards to species characteristics. So what did we actually find? We go through all of these best practices to make sure that we're going to be able to estimate phenology at a really broad scale across many years using messy data, and we want to make sure that we are able to find results that make sense. And our case study showcased that there's this unique power of natural history collections data to test existing hypotheses. So one thing that has been shown in the literature is that spring and summer species are advancing their phenology earlier in warmer years. And what we find is that across a temperature gradient, as the temperature gets warmer, spring species that fly in the spring and the summer do indeed fly earlier in warmer times and warmer areas. But fall species have often been shown to either delay or not advance their phenology, and we find that same thing. So I think we're able to use this big data set to confirm existing hypotheses across even more species and greater distances. And we also found that the end of the flight period to be later in warmer regions, and this was especially true for multivoltine species, which makes a lot of sense. Species that can add broods across their range if it's warmer are going to do so and extend when they stop. But getting back to our research questions, so we were able to have a spot check that we're finding expected results. So what do we find in areas where other research has not been able to ask these questions across really long time frames? How do the phenologies change through time after accounting for climate? Well, we found that we were able to generate new insights about temporal phenological trends. Specifically, we found migratory species and species that enter diapause as adults are advancing the start of flight periods in more recent years, even after accounting for climate context. These results highlight the physiological and adaptive differences between species with differing overwintering strategies. So after we accounted for changes in temperature, changes in precipitation, and the seasonality through all of these years, there was still something that we are unaccounting for in our temperature, annual temperatures, that is leading these species to have earlier beginning of the flight periods, and species that are overwintering as larvae or as adults or are migratory show these really different patterns. So there's more work to be done to understand the drivers of phenology in those groups of species. We also found that those species were having different responses through time of when they were ending their flight periods. So migratory species are beginning to migrate out of their summering grounds earlier in more recent years, even after accounting for climate context, whereas species that overwinter as adults, they are ending their flight periods later in more recent years, even after accounting for these climate contexts. So again, for these two groups of species, there is some sort of climate or other variables that we are not accounting for in the model that is changing through time. That is an important aspect to their phenology that is not included yet in our models, but understanding what these drivers are will be really important to be able to predict their response to global climate changes in the future. So as demonstrated in my talk, working with digital natural history collections data takes expertise in the biology of your study organism. You also need expertise in data science, ecological modeling, and an understanding of how specimens are collected and stored. It may be tempting for one to be a sort of a Swiss Army knife and take on all of these roles. However, as anyone who has ever tried to use a multi-tool can opener, so using one of those tiny can openers, you know that there are better tools for the job. And likewise, assembling a team with cross-cutting expertise will lead to better and more interesting science. Thus, I view partnerships among data scientists, museum experts, and ecological modelers as the surest route towards new innovations that can further harness natural history collections to address pressing global change. My hope is that continued sharing of these innovations across these disciplines, aligned with clear descriptions of best practices, to help to fully unlock our understanding of the tempo, mode, drivers, and outcomes of phenological change at the broadest spatial, temporal, and taxonomic scales. And I have one more sneak preview about work that we've been working on that I want to talk about before ending the talk. And that is, we just talked a bunch about how overwintering strategy is important to phenology. And that has been known in an absolute sense. And we just demonstrated that overwintering strategy is also important in an interaction between that trait and time. But does overwintering strategy also influence abundance trends? Phenology offers a way for a species to adapt to climate change. Climate change is known to impact butterfly trends, so you might expect this trait to also be important to abundance trends. So this is work that we're still working on, but I have some results I will share with you. And this includes that we were looking at a recent paper that Michael Crossley and colleagues published, where they found that recent climate change is creating hotspots for butterfly increases and decreases across North America. They found that in areas with increasing precipitation over their study period had species with greater increases in abundance trend over years, and species in warming areas experiencing global warming, we're finding abundance trends that were decreasing. This paper also found that there were no traits of the traits that they included in their study that were really predictive about which species were the winners or losers of these changes in climate. However, they did not include overwintering stage as a predictor. So I did a reanalysis where I used their data, but all for each species in their analyses of butterflies, I added in their traits of overwintering stage. And what did I find? I found that traits were important in predicting these abundance trends, and the overwintering stages in areas of temperature that were warming or warmer areas, adults were found to have the greatest declines, and also species that overwinter as eggs in these warming areas. The other groups had about flat trends. So that trend we saw with a negative response to temperature seems to be driven primarily by species that overwinter as adults and eggs. The increase in precipitation seems to be with increasing precipitation and increase in abundance trend, the overall trend showed by Crossley et al. That seems to be primarily driven by species that overwinter as adults, are migratory, or overwinter as, or not, are larval overwinterers or overwinter as pupa, whereas species that are migratory or overwinter as eggs actually show this decline in abundance in areas with greater precipitation. So in conclusion, my work is showing that overwintering stage and its interaction with climate is critically important both in butterfly phenology and is appearing to also be important about abundance trends, and they differ dramatically according not just to overwintering stage, but also its interactions with climate. I want to thank all the individuals who contribute to making specimen records freely available, and that includes the volunteers who help in museum collections and also digitize those data, and also people who are spending their time uploading to sites such as eBird and iNaturalist and other citizen science data sets, or making identifications on those platforms. Without that, really, a lot of my work would not be possible. I also will briefly mention that these work have been funded by the National Science Foundation and the UFBI. And thank you all so much for attending. I look forward to answering any questions.
Yeah, that was a superb talk, Michael. We got engaged from slide 1 to last one. And anyone has any question, they can ask. They can unmute themselves and they can ask if they have any question. Okay, if we'll wait for some people if they want to ask, or they can also type in the chat box. So till that, we have one question actually. That is,
Okay.
you showed that that flight and nectar phenology, those graphs. Yeah. So how we can do that? How to proceed for those types of studies?
Yeah, absolutely. Those types of studies, I was doing intensive field work on those. So I was spending, because that species flies so has such a short flight period, we were able to basically go out in the field, myself and others, every single day, where we would walk along the same transect, and we had transects already laid out. So we would walk around the same straight line, and we would count the number of butterflies we saw at different points, and at different points we also had a nectaring count. So we would count actually along the transect as we walked the nectar sources, and then at each point we would stop to count butterflies. So that took really intensive repeated sampling of the fieldwork. There is some ways to potentially automate those recordings using like photo trapping. For butterflies, it would be harder. That probably will take people going out looking at it. But for plants, there are increases of photo-based approaches where you take a repeated photo every day or every other day, and then you can count how many things are in flower or the green up, stuff like that. So a more remote sensing approach.
Okay, that's really a huge amount of fieldwork needed, as far as I just got to know.
Yeah.
Yeah, and so after we take all those after the field work, so the main thing which comes into my mind, that is the analysis part, and analyzing these huge data. Actually, it's a very very complicated thing, actually, I guess. So how to approach analyzing these types of data? In some small steps, if you can say, to all those young researchers who have joined here, I think they would really want to know about that, analyzing those types of huge data sets.
Yeah, totally. I think that the most important thing to have when you have big data sets is to actually start with a good question. And there are so many different ways you can use these data for. And I think sometimes people are tempted to just try to start playing with the data, and then they start asking 100 different questions. And that becomes really hard to figure out the best way to answer 100 questions. So before you start working with the data, I think the most important thing you can do is come up with a really clear ecological or evolutionary question, a biological question that you want to answer, and then think of your study design and how you would want to do that. So that would be step number one, is think of your question and study design. And then number two goes into the more computational work, where I think it's breaking it up into cleaning your data, understanding how to use the data. So that would be a lot of data visualization, I think. It's hard to know all of the potential problems with the data unless you try to plot it. So figure out where these records are found, on what days it's found. And by doing that, you might figure out the different issues. So looking at data visualizations in a bunch of ways is helpful to understand the data. And the majority of the time in big like data projects is really spent cleaning the data. So if it feels like you're spending a long time looking at the data and cleaning data, you're probably doing it right. The analysis stage can be very computationally expensive, where your computer is doing a lot of work, but usually has less human input, I would say, on these really big projects.
Yeah, we should actually think for the topic first, then we should, and then go ahead with the type of analysis that would be appropriate for that data set, right?
Yep, exactly.
So yes, so someone has put up a question in the chat box. Yeah.
Yep, I wanted to know which software is used to analyze the data. And I would say about 95% of the time I'm using R. So I use R for most of my data science, visualizations, GIS. Most of my work is used in R completely. Sometimes I will use SQL, and oftentimes I'll use SQL through R if the data is so huge that R is being super slow. I'll use SQL, and then Python will be used occasionally too, if I ever need machine learning based approaches. I will move to Python, but about 95% of the time I'm working in R.
Okay, so we have another question from Ritham. Okay, sorry, another question has just popped up. So that is, what career options do we have in biodiversity? Yeah, for a 14-year-old.
That's great. Yeah, I think there are a few different options for sure, and that would involve there are like more academic routes, which is what I'm doing, where I'm within a university system doing research, and that provides lots of opportunities from evolutionary questions, ecological questions, or conservation questions. But then there are also going to be biodiversity careers where you can work for non-governmental organizations, nonprofits that are interested in conservation or other land management based approaches. And that could be from a data side where you're working on data or mapping for those. They also need a lot of people doing field work. So there are field-based approaches for biodiversity science where you're collecting data. And there is a lot of opportunities too for communications and education outreach. So getting more people excited about biodiversity, whether that's through field trips or going into classrooms. And that can be done either through like more traditional schools or nonprofits as well. And then I think two other opportunities would be through potentially some federal agencies. So in that could be working for like national parks or other sort of provincial park services where they're interested in knowing about the biodiversity, or other management agencies that manage land. And there are also a few industries where, especially for dragonflies, that's a really great one, dragonflies and damselflies and other aquatic macroinvertebrates are great indicators of stream health. So some industries actually use and collect macroinvertebrates to measure stream health. And so you can get jobs collecting macroinvertebrates and then identifying macroinvertebrates that are measuring stream health and looking at how the different companies or pollutants may be affecting that. I think great ways to just get into it is to try to find others or volunteer or talk to people about the opportunities, and always happy to chat more about it too. Dragonflies and damselflies are becoming a my first love is definitely butterflies, but I am becoming a second love for dragonflies and damselflies. They're so cool.
Okay, so we have another question. That is, have you find any shifting of choice in nectaring plant in light of climate change studies, as you suggested in the your slides? So have you find any shifting of the choice of nectar?
I have found shifting of the choice of nectar, and I don't know if it's climate change exactly related or other changes. So for the Pauschik skipperling, that endangered butterfly species, it occurs in prairie fens. So these wet prairies that are really have complex hydrology. And it's possible that there's been, due to agricultural practices, there's way more nutrients that are coming into these prairie fens now from like agricultural runoff. So it could be due to land use change that these nectar sources are being changed. But I did a big historical data dive where I tried to find every historical record of those species. And in doing so, I learned that this nectar plant, Lobelia spicata, was its most preferred nectar plant historically in like the 70s and 80s. That's where this butterfly was preferentially nectaring on. And in areas that the Tausig skipperling occupied when I was doing my study, I didn't see that plant at all. I found it in other spots of the fens, but not where the Tausig was occupying. So yeah, there were documentation of shifts in nectaring, and that could be to climate change or other land use changes, but it's absolutely an important aspect to consider.
So that area is still open to explore, right?
Yeah, absolutely. And the consequences of that is really open.
Yeah. So all the young researchers here, you guys can take a note that these are the open areas for you guys to explore. Yeah. So another question that is more towards the like the data that you have got from GBIF, the data that is imported there using some social platforms like Facebook or something like that, where the people post their observations of different species, right? So
Yep.
so how to get that, how to score the authenticity of those observations, or say how error prone those observations can be, how to include that in your analysis. But that, yeah, we have included this particular something like, so that we can avoid this error, some index to
Yeah.
avoid the error or something like that. How it can be done?
That's a really great question, and is an area of open research that we're still trying to figure out. But there are a few ways that people can tackle that. One is it probably depends on the taxa. So certain groups are going to be more air prone than others, and those are going to be related in a lot of ways to how many experts there are in the area, how potentially closely other things look at it. So there are some butterflies that are, in general, butterflies, especially in the North America, are pretty well studied. And a lot of them can be ID'd easily. But there are a few species, especially in like the genus Beloria, for example, that can be hard to ID. So I would be more skeptical about using some of those data compared to like Papilio, the genus of swallowtails, for example. And ways that some people have been doing to look into it is trying to quantify error rates. So that can be looking at a subset of data and then having experts score that and incorporating those error rates into your model. And other ways to do that is actually, there have been people, especially when you have a whole lot of data, you can afford to do approaches like this. And it's been really common in eBird data, and eBird is pushed to GBIF. They score the actual like observer expertise. So you can, based off of like how long their checklists are or how many species they find on a checklist that you would expect to be in the area, you can score each observer on an expertise level. You could do a similar thing with museum records where you have expertise levels based off of who's identifying the specimen, and then you could especially either use only the records with a certain amount of expertise, or you could downweight records in a statistical framework, give them more uncertainty if there are lower expertise on certain observations or identifications.
Okay, yeah, that really is very important, I guess, for analysis part. These small things, but they are very significant. They can be a significant role. So now I think we can accommodate just one question, last question. So
okay.
Yeah, oh, just one question came up.
Yeah, I love this question. The question is, you have worked on shift of insect phenology in response to global change. Most of them interact with different plants. Have you checked the trend of phenology shift in some of those associated plants to synchronize the plant-insect interaction? And this is something I do want to do more explicitly, is tie the shifts of like a host plant shift to butterflies, or if there are more nectar specialists, the flowering time of the nectar source to specialists of a butterfly or moth or bee. And to date, I have not tied those specifically. When I've looked into that, of the species I had interest of, there just were not enough observations to look at that through time. But as data becomes more and more available, that is something I'm interested in. But also, there are so many different ways to do that. That is a great open area for research. The smallest aspect I've done on that is some work that we have in prep right now is looking at shifts in butterfly, adult butterfly timing, and how sensitive that is to temperature, shifts in plant green up using satellites to estimate when plants are greening up through time and through space, and then also the shifts in the fledging timing of birds through time and space. And we find that plants and insects, or in this case, the green up of forests and the timing of butterflies, are shifting in relatively the same ways to changes in temperature, but birds are shifting way differently. So plants and insects are similar, but birds are different. So there could be actually a chance for phenological mismatch or asynchrony between birds and caterpillars. But that is at really a community level. The question, I think, is really a species specific, and when there's mutualism between plants and insects, and I have not done that, and I think that is way interesting. I would love to do that, and it's a great spot for other people to be researching too.
Yeah, you explained it really well. I guess many guys will be interested to explore this particular area. It's a very interesting one actually. So this sums up our today's session. So it was a wonderful talk that you gave, and it was so much interesting to see all those different some increasing in hotspots, some decreasing in hotspots. Those shifts, dynamic shifts, they were very interesting. So we look forward to have some other talks with you in the future. And for other participants, we stay tuned for other talks of these series in coming weeks. We'll have multiple talks. So by this, thank you, Michael, to be with us today. And
yeah,
and thank you everyone for joining us. So we'll log off right now. So do you want to say something?
I was just going to say thank you very much for inviting me. I had a great time, and these were really great questions.
Yeah, I look forward to meet you again. Yeah, sure. Thank you, everyone.